as a stimulus factor for the production of things such as activated protein kinase and other NFKB. These substances are really important to have in balance. You need to have just enough of those reactive oxygen species to stimulate important components in gene transcription. The acetyl-CoA is also important for just looking at histone acetylation reactions, again, within our DNA, that gene transcription. So there's this back and forth communication between our mitochondria and our DNA held in the nucleus. It isn't just a one-way street, but the balance of those reactive oxygen species that you see on this slide, which are a stimulus for some favorable changes, it's the amount of that reactive oxygen species pool, that quantity, which can either make or break us. Excessive amounts of these reactive oxygen species, because we don't have adequate endogenous antioxidants being produced, or we don't consume enough, let's just say, vegetables and brightly colored fruits and spices and things along those lines, which are rich in antioxidants. An inadequate quantity of antioxidants available to the mitochondria will ultimately bring about their demise because of these reactive oxygen species. So it's always a balance, and it's really important that it be discussed that way. In the case of sarcopenia, these excessive reactive oxygen species are one of the more primary causes for the loss of viable skeletal muscle, which has to be supported by mitochondria. So if we don't have enough mitochondria in our muscle, eventually that muscle disappears. That's really what we're talking about with muscular atrophy, and that which typically occurs later in life. Often it's associated with things like a vitamin D deficiency, inadequate protein, obviously inadequate weight-bearing exercises or strength training. All of these things are important, and they're all a facet of sarcopenia if they're absent. But the presence of excessive amounts of reactive oxygen species overwhelming the mitochondria through inflammatory reactions is a prime driver of sarcopenia. So that's just, again, this is one example of where a loss in mitochondrial function is going to really affect a particular type of human tissue or organ. When it comes to renal health, we know that cystic kidneys have a very unique quality, or the absence of mitochondrial function. So these afflicted kidneys tend to be totally dependent on glycolysis, which is how we run glucose for a modest amount of ATP without using the Krebs cycle or the mitochondria. That's actually called the Warburg effect. It was discovered by Dr. Warburg back in the early 1900s. And we know that it's not really just with respect to cystic kidneys and renal disease. We know that it's also a primary cause of most forms of cancer. And that's why oncologists who are studying this, as well as researchers such as Thomas Seyfried at Boston College, are making a strong case for cancer to be looked at as a chronic disease or a metabolic disease. And what this slide depicts is how mitochondria, when damaged, which you can see on the far left-hand side, by a variety of conditions, whether those be oncoviruses, carcinogens, that damaged mitochondria also causes the same thing we talked about in the previous slide, which is an over-reliance on glycolysis, which is where cancer cells really start to generate the vast majority of their energy, and that the progression of tumors and cancer in general are much greater when you have lower levels of this oxidative phosphorylation, lower levels of mitochondrial energy production, or beta oxidation. So, it's really interesting to see all of the different conditions and diseases, but they have this core similarity, which is where the mitochondria are not helping a particular tissue, organ generate enough energy to be functional, or in the case of cancer, it's completely absent. And then there are chemical pathways which are initiated by an over-reliance on glycolysis or carbohydrate metabolism that really lends itself to angiogenesis and vascularization of a tumor. So And I'm going to share with you throughout this discussion some excellent papers that I would encourage you to look at when you have an opportunity that really take a deep dive examination of mitochondrial dysfunction and its connection to different types of symptoms and conditions. One of the better ones is by Morris and Burke, and it looks at the different ways in which mitochondrial dysfunction arises. And we're going to cover those this afternoon. We're going to look at 3 or 4 different primary causes or mechanisms involved in the pathology of mitochondria. This is an excellent paper that discusses those. One of the most important ones is the overall abundance of highly peroxidizable or oxidizable lipids. And these are highly polyunsaturated fatty acids. In particular, the omega-6 fats, a loss of membrane integrity or an excessive amount of mitochondrial membrane permeability, leaky excessively permeable mitochondrial membranes. We've already briefly discussed an excessive amount of reactive oxygen species in relationship to the endogenous antioxidants that should be there. And then there are specific toxins that can also really knock out mitochondria because of some of the unique effects they have on biochemical pathways within that organelle. Now, these are those 4: lipid peroxidation, especially when it comes to the industrial seed oil, omega-6 fatty acid rich fats. You see over the course of this talk, and you'll see in my recommended reading or references toward the back of this talk, I'll highlight some of the better papers that look at omega-6 fatty acids and their influence on mitochondrial health. And across the board, they're damaging. We know that they contribute to inflammatory pathways in the body. And we know that the average American diet, and for that matter, the average industrialized modern diet, is excessive in its omega-6 fatty acid content and very low in its omega-3 fatty acid content. So this is a very consistent theme throughout most examinations of chronic disease, and it's no exception. Mitochondrial health is by and large determined by a few factors, dietary factors, and the types of fatty acids present in any population's diet is often reflected in mitochondrial health later in life. Excessive reactive oxygen species, again, they're going to be an inherent part of mitochondrial metabolism. Anytime you are producing ATP, there will be these reactive oxygen species generated, and that's okay. What's important is that the body and that cells have adequate amounts of endogenously produced antioxidants. One of the big ones is superoxide dismutase, SOD, and there are different forms of that. Some dependent on zinc and some more dependent on manganese. Then there's glutathione peroxidase. These are examples of these antioxidants that we produce in our body from specific trace minerals, having adequate amounts of those trace minerals, having adequate amounts of protein and certain amino acids. And those are the most powerful antioxidants we have in our body, the ones which we produce naturally. But then, of course, you get added benefit from the antioxidants found in dark green vegetables and in spices like turmeric and in certain fruits like berries and citrus peels and things along those lines. So there's this, again, there's this balance that we have to have. Physiologically, we have to have a certain amount of these reactive oxygen species. We can't have inadequate, but we can't have an excessive amount. And then we have to have the antioxidants to balance those out. And when there's an imbalance, when we're generating large amounts of these free radicals, in essence, or reactive oxygen species, we're going to damage healthy tissue. Environmental toxins, there's pesticides and agrochemicals such as herbicides and things along those lines, which are clearly damaging to mitochondria. And we can see that with an investigation that each and every one of those pesticides, which have known effects on neurological health in particular. And then there's mycotoxins, which are produced by mold. We know there are bacterially derived toxins, such as those which are produced by blue-green algae, cyanobacter. We really see the link between cyanobacter exposure, the toxins produced by those bacteria, in Parkinson's and ALS, where the mitochondria are really knocked out by those toxins. And those individuals that suffer from those conditions tend to lose a large percentage of viable mitochondria over time, with those toxins being produced either from the GI or from their environmental exposure. And then, one that is, I think, most interesting because of how it connects fructose consumption to all of these different types of metabolic diseases that we see people being afflicted with at an unprecedented rate. And knowing how much fructose is in the average, again, person's diet living in an industrialized world, we can look at the metabolites of fructose metabolism. And again, fructose is very different than glucose and other carbohydrates. Fructose requires phosphorylation. So we have to take some phosphorus off of an ATP molecule and attach that to the fructose. And that whole chemical pathway or process generates these advanced glycosylated end products, one of which is known as methyl glyoxal. That's really damaging to cellular health, especially the mitochondria. So we'll take a look at that. We'll really look closely at fructose metabolism, what's different about it, and how methyl glyoxal may be a driving force behind so much of the mitochondrial pathology. So these are the mechanisms. And the first one we're going to look at is the high level of omega-6 fatty acids and how that relates to this. We know that the average American has a ratio of somewhere 30-1, maybe it's 32-1, but it's in that area. It's very consistent depending on whose research you look at. And we know that over the course of human history, left to more of an ancestral diet, where there's an absence of these more modern industrial seed oils, the ratio has always been consistently 3-1. Never higher than 4-1, in some cases maybe 2-1, but it's 3-1 on average. And we know that it might take a ratio of 1-1 to actually reduce inflammation, reverse some of those processes once the fires really, once they catch. And if we look at some of the more common oils used in food products, ranging from food products for the critically ill and medical settings to those which are maybe a part of snack foods or school